So today we'll be explaining M phase, starting with prophase. In early prophase, the chromatin begins to condense to form chromosomes. And the chromosomes are held together in the center by a centromere. The chromosome will later become two different chromosomes later. As the chromosome condenses, the nucleoli begins to disappear. And as the nucleoli disappears, the centrosomes located in the cell begin to separate because of the formation of the meiotic spindle. As the meiotic spindle forms, the centrosomes are pushed to two separate poles of the cell, and this formation is called the aster.
The late prophase is the site where the two sister chromatids get prepared for division. At that point, the nuclear envelope breaks out and allows the spindles and the chromosome to interact. The growing spindle microtubula get attached to the kinetochore and form special protein called kinetochore microtubula. The spindle that did not get attached to any chromosome are called the polar microtubula. The kinetochore microtubula attach to the chromosome and pull them to the pole and allow them to center in the middle of the cell.
Hello, I'll be introducing metaphase. So in metaphase, the centrosomes are at the opposite poles of the cell. And then the chromosomes line up in the middle of the cell, where with their centromeres aligned slightly in the middle of the spindle, where they create an imaginary plane known as the metaphase plate. Then the enzymes act together to separate the chromatids apart, leading us to anaphase.
Hi, I will be introducing anaphase. Anaphase is the shortest cycle in mitosis. In this process, the two sister chromatins split and become their own separate chromosome. Also, in this process, the kinetochore attached to the chromosome, pulling them towards the poles that the chromosomes are facing. As well, the polar microtubula slide past each other, lengthening and basically pull apart the chromosome. As an end result, the chromosome resemble a V. Now introducing telophase.
Hi, telophase is the final phase of mitosis. It is said to be just like prophase but in reverse. Each of the chromosomes line up at the opposite end of the cell, uncoiling and returning to its chromatin thread-like form. At that point, the spindle fibers break down and disappear, and each daughter nuclei is identical to its mother nucleus.
Cytokinesis. That's the one I'm going to be talking about today. So, cytokinesis is starts during the anaphase, and it is completed during the mitosis ends. A contractile ring made of actinic filaments draws plasma membrane the active filaments forms the cleavage furrow over the top of the both cells. The furrow then deepens until it pinches the cytoplasmic mass and separating it into two different parts, yielding two daughter cells. Those two daughter cells are typically smaller and less cytoplasma than the mother cell, and it's also generally identical to it. Now, Tara and Cassie will be explaining the difference between mitosis and meiosis.
Mitosis produces cells identical to itself, which is responsible for the growth and repair of cells such as skin cells, while meiosis produces cells with half the number of chromosomes because they're responsible to produce sex cells such as sperm and ova.
Another difference is that in meiosis, crossing over occurs, and that is the mixing of chromosomes, which allows for haploid cells to be made. And in mitosis, there's no crossing over because it's just a replication of the same cell. Also, in meiosis, cytokinesis occurs twice in telophase 1 and in telophase 2, while in mitosis, cytokinesis only occurs once. In the M phase, cytokinesis only occurs once because it's only one cell division. And now they will present the passage through of a protein through the cell.
Hi, I'm back. Now, I would like to talk to you about the rough ER. Now, the rough ER correct name is called rough endoplastic. Now, in that process, the rough ER can be studied with ribosomes. Ribosomes are composed of two subunits, which consist of RNA and protein. In the rough ER, you will have the ribosome that will be studied in the rough ER. Also, you will have those that are free. Those that are free will be attached with sugar, and then they will go to the vesicle to be transported to the Golgi apparatus.
So as the proteins are pinched off and enter the cis phase of the Golgi apparatus, it is then modified, packaged, and secreted through the trans phase of the Golgi apparatus into a transport vesicle, where it is then transported to its final destination.
And that will be the end of our instructional video today, and I hope it helped you guys out.